Bun găsit la emisiunea Știință și Cunoaștere. Este pentru a doua oară în cariera mea de jurnalist când un cercetător științific laureat al Premiului Nobel este prezent la emisiunea Știință și Cunoaștere. Peste câteva momente îl veți cunoaște pe Jack Szostak, cercetător la Institutul Medical Howard Hughes, profesor de genetică la Școala Medicală Harvard și profesor de chimie și biochimie la Universitatea Harvard, cercetător la Departamentul de Biologie Moleculară Massachusetts General Hospital. Tema emisiunii este Originea vieții pe Pământ, un subiect despre care vă garantez că va avea un impact semnificativ asupra concepțiilor, credințelor și ideilor pe care le-ați avut până în prezent, precum și asupra întrebărilor existențiale. Cine suntem, de unde venim și de ce există această lume? Ei bine, deși vi s-ar părea că răspunsul e simplu și evident, totuși, din numeroasele încercări de a descifra misterul, începând cu anul 2009, profesorul de genetică și biologie Jack Szostak a reușit să recreeze un model ipotetic de laborator pe baza căruia să demonstreze modul în care substanțe simple, existente pe planeta noastră în urmă cu peste 4 miliarde de ani, s-au combinat în așa fel încât să se obțină primele celule primordiale. Profesorul Jack Szostak a realizat o descoperire inedită și a reușit să sintetizeze cromozomi și alte organisme celulare capabile să autoevolueze singure mai departe. La începutul lunii iulie 2016, domnia sa a acceptat să participe ca invitat la emisiunea Știință și Cunoaștere, înregistrările fiind realizate prin camera web. Jack Szostak, născut la data de 9 noiembrie 1952 la Londra, Marea Britanie, este biolog laureat al Premiului Nobel pentru Fiziologie sau Medicină pe anul 2009, împreună cu Elizabeth Blackburn și Carol Greider pentru descoperirea felului în care cromozomii sunt protejați de telomeri și de enzima telomerazei. Începând din acel moment, atenția sa este focalizată asupra dezlegării misterului apariției vieții, fapt care ar permite omenirii să treacă printr-un salt calitativ de neimaginat. În mod neașteptat, interviul s-a extins la o durată de peste 73 de minute, ceea ce mi-a permis să realizez 2 ediții consecutive, la care am mai adăugat câteva comentarii și completări utile prin prezența excepțională a profesorului de chimie Adrian Pătruţ, care e interesat, printre multe altele, de cosmologie, originea Universului, apariția elementelor chimice și implicit apariția vieții. Așadar, toate acestea văzute din perspectiva chimistului și cosmologului. Fiți alături de noi la acest eveniment de excepție la Știință și Cunoaștere. Dear Professor Jack Szostak, it is an amazing opportunity and I welcome you to our Science and Knowledge TV program for the first time in Romania on TVR Cluj, Romanian television.
Thank you very much. It's my pleasure. Always happy to talk about interesting science.
I'd like to use every second I have for this amazing interview. Please tell us how and when did you and your team started the experiments on evolving the functional RNA in order to create auto-assembling organelles and even a primordial cell?
Well, it's actually been happening gradually over a long time. Over 20 years ago, I first got interested in RNA and the origin of life. I think that happened really because of the discovery by Tom Cech and Sid Altman that RNA molecules can act like enzymes. And so that started to get me interested. And then over the years, we worked a lot on RNA and learned how to evolve new RNA molecules, the things we thought might be more relevant to the origin of life. And then over the last decade or so, we've really become much more focused on these origin of life questions and really directed research in the lab towards trying to recapitulate some of those processes by trying to understand how we could build up an artificial cell, something like the first most primitive cells in the lab.
What is the current phase of your amazing project? Did you actually succeed in creating a self-replicating protocell or a cell-like structure in your lab?
Well, that's what we're working on. We're definitely not there yet. It's hard to say how long this will take. There still are a lot of open questions, a lot of things to do. But maybe I can explain a bit more about where we are. So to do that, I have to explain a little bit about how we think of a primitive cell. And so we imagine the first cells as being extremely simple and consisting of little more than a cell membrane made of fairly simple molecules that can grow and divide. But inside, it has to have some genetic material, which we think will be RNA or something like RNA. And that also has to be able to replicate so that its information can be copied and passed on from generation to generation. So, and everything has to work together, of course. So what we have learned about the first part, the cell membrane, is that there are actually many ways to make very primitive models of cell membranes grow and divide. They can grow in different ways, either by adding more membrane molecules, more food, if you will, or by competition. So some grow at the expense of others. There are different ways we can make them divide. So that part seems to be fairly straightforward. We think those processes could easily happen on the early Earth. The part where we need to worry about the RNA molecules, the genetic materials, replicating before there were any enzymes, right? This has to happen just because of physics and chemistry. That is, seems to be a much more challenging problem or set of problems. So we've been working on that part pretty intensively for the last several years. We certainly feel like we've made a lot of progress, but there's still a long way to go. There are many open questions, many unsolved problems. And then some of the most interesting parts are when you try to get the RNA molecules to work and to replicate together with the membrane that they're encapsulated in, and combining all of these systems and making sure that everything's compatible and one part doesn't interfere with another part. That gives a whole new set of interesting questions. And again, some of those we've solved, some are still open questions, interesting scientific problems to solve. So we feel like we're learning a lot. We're still a ways from actually being able to build a complete protocell model, but I think we're getting closer and learning a lot on the way.
How would you describe the boundary of the transition from chemical to biological? Exactly in what state or phase a supercomplex chemical structure began to behave like a biological structure? Is there something special happening?
That's a really interesting question. The way I think about it is that there's not some particular boundary, right? It was a gradual process of systems becoming more and more complex. Even the process of evolution, which is really central. If anything that is really characteristic of life, I would say it's the ability to evolve, to do Darwinian evolution. Even that starts off at a very simple, almost trivial level, and gradually becomes more open-ended and more able to lead to really interesting new adaptations and more complex forms of cells. So I think there's a whole process starting from simple chemistry, very, very elementary building blocks from the atmosphere or different kinds of mineral ions, leading to a stage of organic chemistry where you gradually build up the components that you need for biology. And then some of these, like the molecules that form membranes, very easily self-assemble into much larger structures like membranes. And that's a process that's been studied and is pretty well understood. Also, once you can make nucleotides, which is something there's been a lot of progress on, but people are still working on it. There's still a lot of open questions. Once you make nucleotides, they can polymerize into long chains, so RNA chains or other nucleic acids. And then you need the right chemical and physical environment to allow those molecules to replicate and also to fold up to make three-dimensional structures that can start to catalyze reactions. And so all of these things happen to make a pathway that leads from really simple chemistry to more complex chemistry to very, very simple biology in the beginning. And then once you can start to evolve new functional molecules, the cell structure can rapidly become more complex. And then we gradually get into the evolution of metabolism and the evolution of protein synthesis, membrane transport machinery, and all of the biochemistry that makes modern life so complicated. So basically, I don't think there's a single dividing line. I think people waste a lot of time and energy debating what is life, or where's the boundary between non-life and life. To me, it's the process, that gradual increase in complexity, that is really what's important and interesting.
This is really amazing, because when I was in school, we were told these are atoms, atoms are forming molecules, and then we make a jump towards something which is alive.
So I think that's a very common point of view, and I think that's maybe one reason why people find it hard to think about the origin of life, because people want to understand how you can go from just some bunch of molecules floating around in solution, and all of a sudden you have a complex cellular structure that's kind of like what we have now for modern life. But I think the only way to really understand it is to imagine a process, a pathway with lots of steps, each one of which is simple enough that we can understand it in terms of the chemistry and the physics that are involved. So there's not one sort of big magical step, but really a lot of smaller steps that lead to finally to forms of life that can really evolve, and then evolution starts to take over and lead to the complexity of modern life.
Can you describe or hypothesize about that hidden mechanism which makes chemical structures to auto-organize themselves into super complex systems? Why they began to move and attract each other at a certain point?
Well, there are actually many simple processes like that that go on all the time. In fact, there's a whole field of chemistry called supramolecular chemistry, where people study the interactions of small molecules that lead them to assemble into larger, more complex structures. And there are many, many examples. But perhaps I could just talk about the one that is most relevant to the origin of life, which is the formation of membranes from very simple small molecules. So in our work, we generally make membranes. They're similar in a way to modern cell membranes, except that they're much simpler and made of simpler molecules. So the molecules we use most in our work are simple fatty acids. And so typically for day-to-day experiments, we use something that's very cheap and easy to get. So we use oleic acid, which comes from the fats in olive oil. If we want to be more realistic, we might use something like capric acid, which is a chain of 10 carbon atoms with a carboxyl group at one end, so a carbon with 2 oxygens. So all these fatty acids have a carbon chain and then an acidic group at the end, a carboxylate, which can be ionized, so it has a negative charge. Okay. So what these molecules do when they're in water is very interesting, because the carbon chain doesn't really like to be in water, right? The carbon chains, the hydrocarbons, like to come together, just like oil likes to form drops and not just dissolve in water. So in order for the carbon chains to come together, but the carboxylate acidic groups to be exposed to the water, then they have to assemble into more complicated structures. And one of those structures, out of several that can form, is what we call a bilayer membrane. So we have two layers. In the carbon chains point to the inside, and the acidic carboxylates point to the water. So you have a surface of negatively charged groups, and then this hydrophobic middle, oily middle, and then another surface of charged carboxylate groups. So these simple, very small molecules assemble into these, initially into sheets. The sheets grow at the edges, and then they close up. The edges have a kind of high energy, and so to minimize the edge, the sheets close up and they form these spherical vesicles with a bilayer membrane. So it's a very beautiful model of a cell membrane, but it forms totally spontaneously from very simple molecules. And that's just one example. There are many, many examples where small molecules assemble into larger structures. And generally, I would say this is because you have opposing forces. So in the case of fatty acids forming membranes, we have this very strong force driving the hydrocarbon chains together, and a very strong force trying to keep the carboxylates exposed to the water. And so these two forces are opposing each other, and the difference between them can be quite small. And if the difference is in the right direction, you form, say, a bilayer membrane. If the difference is a little bit more in favor of the acidic groups, you might form smaller structures called micelles. And if the forces are not strong enough to drive these forms, then you'll just form oil drops. So you have big forces in opposite directions, and the small differences between them control the shapes that are generated.
There were several other unsuccessful experiments conducted over the past 50 years by other researchers, but they never got a plausible path that led primitive cells to emerge from simple chemicals. Please tell us what they tried to do and why the success was somehow limited.
Okay, sure. So I think one of the most dramatic breakthroughs, and really the beginning of this whole field of prebiotic chemistry, is the famous Miller-Urey experiment. So this is around 1952 when Stanley Miller did an experiment where he put an electric spark, a spark discharge through a mixture of gases that he thought would mimic the primitive atmosphere of the very young Earth. And the result was really amazing because this simple physical process generated a huge number of organic compounds. And then when they processed their samples, they were, I think, really stunned to see many of the amino acids that we find in modern biology. So the breakthrough was that what we thought of as essentially biological building blocks could be made in a very simple physical chemical process. Okay, so that was the good news. The problem was that this kind of experiment generates hundreds, thousands of compounds. So you do see some of the molecules that are biological building blocks, for sure. But you also see hundreds and thousands of other compounds that are not part of biology. And it's very hard to imagine how you could select out or use just the small number of molecules that are really relevant to building up biological structures. And a lot of prebiotic chemistry that was done in the following decades, and is actually still being carried out in a number of groups, it is similar in a sense that you take some simple starting materials, some really powerful source of energy, Essentially, you rip these molecules apart into atoms, and you let them come back together, and you make a huge mess of compounds. So the positive lesson, again, is that some of the building blocks of biology can be generated in very simple ways. And studying those pathways has been quite informative. But it's probably not the right way to think about how things really started on the early Earth. So there have been in the last decade or two, there's been an increasing emphasis on trying to understand chemistry that could give you just a small set of compounds. You want to focus all of the material and the energy to generate more or less just the compounds that you need. Of course, we also need to think about ways that they could have been enriched or partly purified on the early Earth and so on. But this is a very different way of thinking about chemistry on the early Earth. So we really need to start thinking about local environments, physical processes that could do interesting chemistry and not maybe in a more controlled way so that you don't generate too many uninteresting compounds. So I think one of the best examples of people who are doing this kind of chemistry is John Sutherland in the UK and some of his students like Matt Powner. Other people have been very focused on this kind of question. So Stephen Benner, for a long time, has been trying to push this idea of finding conditions that will control this kind of chemistry and give you a smaller number of products. So I think we may still be at the early stages of exploring this. This is a whole different way of looking at this kind of early chemistry and trying to understand pathways that could happen on the early Earth that would give us the molecules that we need to build up biology and not a lot of irrelevant material that would get in the way.
The next question is the mother question of all questions. Based on your new findings, how did life begin?
Okay, so of course I don't have a complete answer because there are a lot of parts to that question that are really big and interesting scientific problems. But as I was saying before, the way I think of it is a pathway. And if we actually step back, even from the chemistry and the assembly of the first cells, we can think of the process really beginning with the formation of planets around a young star. So there's a lot of interesting chemistry that happens in the protoplanetary nebula. There are interesting physical and chemical processes that are involved in the assembly of planets. Of course, we need to understand what it's like on the surface of a very young planet. Of course, at the beginning, it's extremely hot, but it will cool down, and eventually you can have liquid water. So there's a lot of modeling that has to be done to understand the development of a planet, the way that a planet changes over the course of its first few 100 million years. We need to understand the atmosphere of primitive planets, the chemistry in the oceans, the origin of land and environments on land. And then we can get to the point of the prebiotic chemistry, the way that some compounds might accumulate with time, form large reservoirs of organic material that could then be processed. And as we build up more and more complicated building blocks of biology, we can start to think about the assembly of primitive cells, the beginnings of Darwinian evolution, when we have cells that can replicate, that have cell membranes and genetic molecules like RNA or something related to RNA. And the process continues as those cells evolve to become more complex and better adapted to their environment. They start to evolve all of their metabolism, protein synthesis, and become more and more complex. And And as that happens, and life becomes more widespread and more abundant over the surface of the planet, life itself starts to change the planet. And so we need to understand the interactions between the whole biosphere and the way that it changes the atmosphere and the geology of a young planet. And that eventually will bring us up to modern life. So there's this whole very interesting pathway that brings together astrophysics, planetary sciences, chemistry, evolutionary biology, and all of the ways that these fields interact. And to me, that's what makes this field so interesting and so exciting, is that to really understand the whole process, you have to bring in scientists, you have to get scientists involved from many different fields. And we all have to learn how to talk to each other. We have to learn enough about each other's fields to be able to start to work together and understand this really big picture of how life started.
Can you tell us more about the cooperation with other scientists in order to push forward your research?
Yes, in fact, we do. We're working on that in many different ways. For example, just locally, we have a group at Harvard where we have people from the astrophysics and astronomy, people from the planetary sciences, people from chemistry and evolutionary biology, and we get together, we have seminars and discussions, we have the beginnings of some collaborative projects. For example, I'm now doing a collaborative project with a group in the Astrophysics Institute. So that took time to emerge, but it's actually very, very interesting and exciting. On a larger scale, there are several programs. One I'm involved with involves the Simons Foundation. So we provide funding to people from different fields. So again, we have astrophysicists, planetary scientists. We're trying to bring in more geologists. We have chemists and biologists. So the interactions and the discussions between these different people are very, very interesting. It's very exciting. And there are many workshops and meetings that also try to really bring together people from different fields. So it's such a big and interesting scientific question that it really does draw people in from many different fields.
Do you believe that it's possible to create an artificial cell that can grow and divide as well as evolve in a Darwinian sense to adapt to its changing environment?
Well, I do. I mean, that is really the focus of my lab in particular. So it's a big, challenging problem, obviously. So we don't expect to get to that point in the next year, but maybe if things go well in the next several years or 5 years, perhaps. I think we have a pretty good handle on the primitive cell membrane. As I said, we can make that grow and divide. We're learning a lot about the process of RNA replication without enzymes, just using chemistry to drive RNA replication. We're gradually getting closer, I think, to being able to do that. We've had considerable progress in trying to make everything work together. So we've definitely overcome some barriers there, but there are other challenges in that area in particular. But I think, I mean, I do think that life started spontaneously on the early Earth. So there has to be a simple way that it happened. Generally, the way things work in my lab, the way our science progresses, when we're focusing on a particular scientific problem, a particular step in this process, at first, we're baffled. We have no idea how to solve it. And then maybe we come up with an idea or a few ideas. We might get a proof of principle solution, something that's maybe not really plausible for the early Earth, but a solution that will work in the lab. Something that gives us a clue as to how to overcome this problem. And that almost always leads to more ideas. And then we gradually work our way step by step to understanding the physical processes or the chemical processes that actually could have worked on the early Earth. So it takes time and a lot of steps to figure these things out. And the ironic thing is when we finally get to a process that we really believe is simple enough and robust enough to work on the primitive Earth, we think, oh, it's so simple. Why didn't we think about this years ago? But you know, it's hard to figure these things out when there's so many possibilities at the beginning. So you have to work your way step by step towards solutions. What's really interesting, I think, is that sometimes we find one solution to a problem, and then we might figure out another way that could work. And sometimes we end up with several different possible pathways that could solve a particular problem on this pathway to life. So I think that's very interesting because if there's multiple ways that something could have happened, it makes it seem much easier. You know, maybe one process could work in a particular environment, but if there was a different environment somewhere else, maybe it could have happened a different way. So there may have been actually multiple ways that life could have started on the early Earth. There's actually a lot of debate. Some of us in our discussions think that, yeah, maybe life could have started in many different ways depending on the particular environment. Others think that, well, maybe the chemistry can really only work in one way. And life actually really had to start with chemistry very similar to what we see now. So the only way to answer these kinds of questions is to actually work out all the chemistry and try to actually build primitive cells or parts of primitive cells, you know, from different kinds of molecules, for example. So we focus a lot on RNA, but there are many other nucleic acids, and maybe some of them could work as genetic materials. So the only way to really know for sure is to make those molecules. First, you have to discover what they are, and then you have to make them. Then you have to try to figure out how they could have been made on the early Earth. And then you have to figure out through experiments in the lab, well, could they be replicated? Could they fold up to make three-dimensional shapes that could act as catalysts? Could they do all the things that RNA can do? Now, so far we haven't found anything like that, so that makes us think, well, maybe the first life really did emerge using something like RNA. But there are a lot of possibilities, and so there's going to be a huge amount of really interesting chemistry to do to explore these ideas and to see if in the lab can we make simple life forms that are based on a different kind of chemistry, and if so, does that mean that life could evolve in different ways, maybe on the early Earth, or maybe on these thousands of exoplanets that we know about now. Maybe there could be different forms of life based on a different kind of chemistry, either a little different or maybe a lot different. So exploring these questions is actually a really interesting aspect of this whole field.
But this could lead to a cultural and informational shock for having a new and totally different understanding of the origin of life, which is now purely scientifically demonstrated, and thus many other cultural and esoteric beliefs or religious beliefs are outdated and worn out. Can you comment on that?
Yeah, in a sense, I agree, because if we have a very clear understanding of how life could emerge in a purely natural process, then it's not really necessary to invoke any kind of supernatural explanation. On the other hand, I think, in my experience, is that people who already believe that that's how it happened are not really going to be influenced by some group of scientists working in their laboratories. I don't think that what we're doing is really going to affect people who believe in creationism, for example. Maybe in the long run, what you said could happen, that more and more people will just accept that life is just a part of nature and something that emerges spontaneously. But if that kind of cultural transition ever happens, I think it will be over several generations. It's not something that's going to be an immediate result, for sure.
There is a new emerging branch of biology called synthetic biology. If now we can create a kind of artificial cell-like structures, then the next step would be artificial life forms, artificial human organs, and even artificial human being. Do you believe that somewhere in the future this will no longer be science fiction?
Well, I think the interesting thing there is that people are already using genetic engineering to modify existing organisms. Right? This is a huge branch of science and biotechnology, much bigger. There are many more people involved in this kind of work than in origin of life work. It's actually much easier to take an existing form of life, whether it's a bacteria or a yeast or an animal, and now we have very powerful technologies to modify the genome, and the implications of this are something I think we all need to really think carefully about. There are many really positive applications. For example, the most obvious are in medicine. I think this kind of biotechnology is going to lead to cures or treatments for many diseases that are really terrible and difficult to approach now. I think the other really big and important applications are, for example, in agriculture. So I think that genetically modified organisms for agriculture can have really great benefits if they're used properly. I think this can lead to, in principle, could lead to less use of damaging pesticides, for example, less use of fertilizers, more nutritious foods, if it's done properly. But we really have to think about how to use these technologies carefully and for good ends. And then there are really, I think, very controversial kinds of genetic manipulations like gene drive technology, which are so powerful that they could even lead to the elimination of, say, certain species of mosquitoes, the mosquitoes that cause malaria, that transmit malaria, or many viral diseases. We have it, I think, almost within our power to essentially drive those species to extinction. And the question is that really something that we should do? We could save many human lives by doing it, eliminate terrible diseases, but then we have to think about the consequences of driving species to extinction on purpose. Of course, human activity in the biosphere is already driving one of the biggest extinctions, mass extinctions ever, which is a terrible thing. Do we want to add to this by doing this on purpose? So there's really a lot to think about. There are very important science policy questions that need to be broadly discussed and debated. And I think this is really driven by just the amazing rapidity of scientific advances in terms of our power to initially make small engineering changes to an organism, but of course it will become able over time to make larger and larger changes. And we really need to think about what should we do and what should we not do with this kind of technology. So again, I don't think this really has anything directly to do with the origin of life, or even the ability to build very simple cells, because the protocells that we're trying to build are so simple, they couldn't really compete in the modern biosphere. And they're so far away from the complexity of any modern organism that I think they also have, well, no practical applications that I can think of. The practical applications for medicine and agriculture and other areas come from the manipulation of existing modern organisms. And that, as you said, this field of synthetic biology is something that's growing and developing very, very rapidly. It's becoming very powerful.
Well, this idea connects with my next question about medical applications of the artificial ribozyme's ability to identify small molecules that bind specific target proteins. Can you make them fight against some diseases which in present time are uncurable?
Yeah, well, that is definitely something I'm interested in. It's the hope that we could do that. So the biggest new class of drugs by far that have been used to treat a wide range of diseases are monoclonal antibodies. So these are proteins. Antibodies are proteins that will bind, they will recognize very specifically other proteins and either inactivate them or prevent proteins from interacting or affect them in other ways. So it's very powerful. Now the question is, can we also do that with RNA molecules? So there's a class of RNAs that we and other groups learned how to evolve in the lab called aptamers. The aptamers are similar to antibodies in that they can recognize other proteins very specifically and potentially could be used therapeutically. Now, going from having an RNA molecule that can recognize a protein to actually having a useful drug is a very, very long and complicated pathway. There are many problems. People have been working now for, I would say, 25 years to solve these problems. The RNA is not stable in the human body. So we had to understand how to manipulate the chemistry to make derivatives of RNA that would last more than a few seconds. And you want them to have the right half-life circulating in the blood. You want them to be very specific so they only recognize the right target. You want them to have a high enough affinity. In other words, they have to have a very, very strong interaction with the target. All of these kinds of problems are being worked on, have been worked on for a long time. And there are some clinical trials in progress now. So I think in the future, we may see more and more therapeutic applications of RNA molecules. Now, the ones I mentioned, these aptamers, are not catalysts. They're not ribozymes. They just bind. As you said, there are other RNA molecules that catalyze chemical reactions. And so these are called ribozymes. And in principle, they could be even more powerful than aptamers. For example, if you want to eliminate a target protein, you could either bind it and inactivate it, or you could bind it and then cut it up so that it's destroyed. Finding ribozymes that are really good at doing that has been very difficult. Again, I think people are working on it. It's probably a solvable problem. And so again, we may see in the future more medical applications of these kinds of RNA molecules, both aptamers and ribozymes. But developing a whole new class of drugs it's also a big challenge and could be very rewarding. It could be very powerful, but it's a long process.
Please let me change the tape and continue our talks later.
Okay.
În primul rând, doresc să urez bun venit, bun regăsit la Știință și Cunoaștere.
Mulțumesc!
Am realizat un interviu cu profesorul Jack Szostak, care a depășit spațiul disponibil unei ediții și am considerat că este util să difuzăm integral materialul în 2 episoade consecutive și deoarece suntem la partea legată de evoluția și apariția vieții, aș dori pentru început să ne povestești despre un lucru premergător acesteia, și anume formarea elementelor chimice din care ulterior, prin autoorganizări succesive, au luat naștere substanțele complexe care se află la baza vieții.
Am o anumită energie disponibilă în univers. Ce se întâmplă? În univers se creează, din fotoni se creează particule, din particule simple se creează particule compozite, din particule compozite se fac nuclee, se fac atomi, se fac molecule, etc. Fiecare din aceste sisteme tot mai complexe este tot mai sărac în energie. Da? Aceasta înseamnă că prin tot mai sărac în energie, prin formarea unor sisteme mai complexe, se eliberează energie în mediu și universul poate evolua în continuare. Apar molecule simple, molecule complexe, etc. Poate până la viață. Viața este cel mai complex sistem pe care îl cunoaștem, poate cel mai sărac în energie, are cel mai puține grade de libertate. Deci acest proces de structurare, de evoluție a materiei, de la particule simple, particule compozite, nuclee, atomi simpli, atomi tot mai grei, etc., molecule simple, molecule complexe, până la viață, ADN, să spunem, ARN, ADN, viață, etc., sunt forme tot mai sărace în energie și care eliberează energie în univers, prin care universul poate evolua în continuare, până unde nu știm. Noi ne-am oprit la viață. Știm o formă de viață terestră care a apărut acum câteva miliarde de ani, da? 4,1 miliarde de ani, atâta știm. Nu știm mai mult despre această viață, că există viață extraterestră probabil. Dacă a mers mai departe evoluția universului sau nu în alte locuri unde viața apărut mai timpuriu, nu știm. Spre ce poate evolua. Dacă sunt sisteme și mai complexe decât viața inteligentă, decât omul și așa mai departe, s-ar putea să existe deja.
Păi, având în vedere vârsta Universului și faptul că sistemul nostru solar s-a format destul de recent, cu ghilimele.
4,567 miliarde de ani aproximativ, dar în orice caz, apariția vieții presupune vieții complexe. Nu a vieții simple, presupune elemente și mai grele decât fierul. Așa înseamnă că trebuia ca o primă generație de stele să explodeze sub formă de supernove și pentru aceasta trebuia câteva miliarde de ani. Pentru a se forma elemente mai grele. Deci trebuie să fie o stea de generația 2, 3, 4, 5, nu știu cât, care să aibă în jur de planete pe care se poată evolua spre forme complexe de viață. Deci n-a apărut foarte rapid, a apărut la câteva miliarde de ani după Big Bang viața în univers, cel mai devreme posibil. De aceea a apărut afirmațiile lui Sagan că noi suntem copii ai stelelor, pulbere de stele, da? Undeva. Este și o carte lui Asimov în acest sens, Pulbere de stele. Iar că suntem copii ai stelelor, deci practic noi suntem formați din colbul stelar, al unor planete care au apărut în interiorul unor sisteme planetare, în jurul unei stele, care făcea parte probabil dintr-o galaxie. Aceasta a explodat ca și supernovă, a aruncat alte elemente în jur și așa mai departe și din acest amestec s-a condensat nebuloasa care a generat sistemul solar, soarele s-au desprins pe rând planetele de tip jupiterian, planetele de tip Terra sau așa mai departe, deci planete formate din gaze sau planete cu o crustă solidă pe care se poate dezvolta viața, da? Deci erau cândva ipoteze restrictive prin care se spunea că omul este o ființă unică în univers, ceea ce pare absurd, da? Pare absurd. Dar nu avem dovezi palpabile, concrete pentru existența, nu a vieții, să spunem, fiindcă îți mai aduc și meteoriții, urme de viață, nu mai sunt în ele, deci viața există probabil în foarte multe locuri, dar de viață superioară, de viață inteligentă. Astăzi avem din meteoriți fragmente, avem și alți aminoacizi decât ai noștri, cu altă polarizare, etc., știi? Deci aduși de meteoriți. Deci știm că există cel puțin cărămizi ale vieții în multe alte locuri din univers. Și acele molecule interstelare, astăzi e clar că premizele vieții sunt răspândite undeva și că atunci când o planetă străbate o astfel de nebuloasă, acei germeni ai vieții pot pătrunde pe suprafață sau în interiorul ei.
În conferințele sale, profesorul Szostak a prezentat o clasificare pe un fel de desen schematizat, și anume că ar exista, să zic, niște etape la modul formal, din punct de vedere didactic, stabilite cu cum a evoluat viața structurată, viața și materia. Și pornește de la epoca formării întâi a Pământului, după aceea epoca stabilizării oceanelor, pe urmă chimia prebiotică, lumea pre-ARN, lumea ARN, lumea ADN, până la deja forme de viață celulară și...
O discuție a unui biolog, fără îndoială, din punct de vedere al unui biolog. Istoria Pământului e mai complicată undeva, fiindcă el a fost o planetă supusă foarte mult timp unor bombardamente teribile, în care n-ar fi putut exista forme de viață superioară. Se spune astăzi că, deci ar exista astăzi dovezi în favoarea faptului că imediat după formare Pământul inițial s-a ciocnit cu o altă planetă de mărimea lui, ceva între Marte și Venus, și că din această coliziune dramatică s-a născut un Pământ 2, fragmente s-au răspândit în spațiu și așa s-a format Luna, care se îndepărtează încet de Pământ. Da? Și că au fost coliziuni fantastice cu meteoriți foarte mari în universul timpuriu, că acești meteoriți erau foarte mulți în sistemul solar, ei s-au mai rărit cu timpul, dar sunt și marile cratere care lasă astfel de urme. Știm astăzi că viața s-a resetat de câteva ori în urma unor astfel de catastrofe cosmice, da.
Cred că dintre toate viețuitoarele, omul este cea mai sensibilă și firavă ființă, pentru că este extrem de sensibilă la cele mai mici perturbări ale mediului, ale patogenilor, deci, și devine din ce în ce mai sensibilă pe măsură ce s-a desprins de mediul natural.
Fiind tot mai specializat, este tot mai sensibil și sunt condiții de mediu foarte stricte în care o civilizație se poate desfășura. Și este o discuție interesantă modul în care un accident, fie el cosmic, fie un accident nuclear major sau așa mai departe, va influența soarta omenirii și cum se va putea adapta omul la noi condiții de mediu. Probabil că foarte greu.
Dacă revenim la descoperirile lui Michael Fossel, de exemplu, prelungirea vieții cu ajutorul unui activator ar putea să schimbe întreaga structură socială, economică, politică, militară, ar putea schimba totul. Și el nu e singurul care are rezultate, pentru că sunt pe cel puțin 3 continente încă 10 cercetători din mulțimea celor care au decis să iasă public și să își uploadeze conferințele pe internet. Pentru că de restul eu nici nu cunosc, dar cercetările s-au extins deja foarte mult. Și ele încep să convergă, ceea ce face Szostak cu ceea ce face Fossel și alții deja încep să convergă și oferă rezultate într-un mod atât de rapid încât nu cred că vor fi interzise.
Probabil că nu vor fi, dar vezi că marele public nu mai face față, nu mai este interesat și nu mai face față. În primul rând, educația nu mai este cea de odinioară, din școală până în universități. Educația este ultra specializată și nu mai este educație generală. Se spune că nu este bine să cunoști prea mult, fiindcă dacă tu cunoști prea mult, poți să ai păreri și poți să fii împotriva regimului politic și la măsurilor care se iau. Deci nici regimul nu încurajează această cultură generală și oameni cu spirit enciclopedic care să aibă un glas în cetate. Da? Deci oamenii de știință au devenit pur și simplu prestatori de servicii. Astăzi, într-o lume dominată de știm cine, da? De negustori.
Da, chiar și așa.
În bună măsură,
ei, la rândul lor, privatizându-și cercetările și având fonduri pe durate mari de timp, vor obține rezultate.
Fără îndoială.
Încă Fossel are compania lui privată. El cu încă cineva au firma, s-au retras dintr-o altă companie.
Va depinde modul în care aceste rezultate vor putea fi implementate la scară mare și aici trebuie și un sprijin al politicului undeva. Știi? Deci e o poveste de viitor.
Păi, mai devreme sau mai târziu se va ajunge, pentru că dacă nu va fi farmacogenomica care o ia înainte și vine cu tratamentele personalizate și strict pe genom și aceea poate să ajute la mărirea speranței de viață chiar foarte mult.
Da.
Pentru că atunci nu mai ai efectele medicamentului clasic care este redus de 10.000 de ori ca și dozaj, va fi foarte bine țintit, va rezolva problema în timp foarte scurt.
Pe asta e medicina viitorului, da? Deci va fi o medicină direct țintită, da?
Da,
Pe
da.
cromozom, gene, etc. Și va fi o medicină de accident, traumatică, pentru accidente. De urgență, atât.
Discuțiile cu domnul academician Popescu, chiar la un moment dat puneam așa metaforic că această nouă medicină personalizată ar fi ca un fel de homeopatie științifică, aplicată științific.
Da,
Deci dai dozaj foarte mic, dar pe baze științifice.
Da, depinde care va fi riscul, de exemplu, rezistenței corpului medical sau așa mai departe, sau farmaceutic la așa ceva. Știm, de exemplu, când eram student, știam că sunt produse ceramice care se pot folosi la obturații dentale și care rezistă mii de ani. Ce s-a întâmplat? Le-au cumpărat stomatologii și le-au închis pentru totdeauna, da? Ce ar face stomatologii dacă ar pune plombe definitive? Da, este așa, totuși a venit la implant, dar se fac multe lucruri noi și în domeniul stomatologiei. Este un progres foarte mare față de ce era acum decenii.
Plombe de 25 de ani și care n-au căzut și deci sunt cazuri când și astea clasice funcționează atât de mult încât sunt comparabile cu speranța de viață pe care cel puțin teoretic o mai avem.
Va putea să fie și o rezistență a brestelor. Dar mă rog, în cele din urmă se va impune. Am spus că ceva mai cu câteva frâne, nu foarte.
Deja s-au dat drumul la o mulțime de patente, ai văzut că sunt tot felul de baterii din astea tot mai mici, cu de putere tot mai mare.
Nu mai poți opri pentru că sunt popularizate pe internet. Și atunci află lumea de ele și nu mai poți să le închizi. Adică nu mai sunt niște hârtii care circulă și le-a pus pe internet și atunci unde ce să fac cu ele? Deci e mult mai greu de secretizat ceva astăzi, da? Decât era înainte. Deci asta e o speranță undeva.
Da, și dacă, de exemplu, ar dori să oprească o producție de serie mare cu bani uneori nu foarte mulți, chiar și un om la el acasă își poate construi anumite lucruri. Pur și simplu, ai informația de pe internet, ai toate datele, ai toate concepția, cum se face, cum se construiește și lumea își construiește. Bobinele alea lui Cheșe, lumea e nebunită, le face acum sute sau poate mii de oameni care s-au ocupat să le construiască și le construiesc.
Da, fără îndoială că evoluția este mult accelerată acum. Cunoașterea, într-un fel, este mult accelerată și datorită mijloacelor de răspândire a informației, da? În principal internetul.
Adrian, îți mulțumesc foarte mult pentru faptul că ai participat la Știință și Cunoaștere și vă mulțumesc și dumneavoastră pentru faptul că vizionați Știință și Cunoaștere în fiecare sâmbătă și duminică de la ora 18:30 pe TVR Cluj sau pe internet și vă invit data viitoare la o nouă întâlnire cu Știință și Cunoaștere.